Welcome to Chapter 7 of Biomedical Physiology. Here we're giving you an intro to endocrine system, the control mechanisms that surround secretion in order to prevent too much secretion or too little secretion of hormones into the bloodstream affecting target cells. There are several topics in this chapter, of which we're only going to cover 3. So we're going to get rid of those, and what we're left with is categories and what constitutes a hormone, how we classify them, and a few examples of release control, particularly what we call the axis. That's one of the more classical control forms, but it does a really nice job of showing examples of negative feedback and some of the complexities therein. One thing to note about hormone disorders and endocrine-related pathologies. The syndromes in hormone and endocrine diseases mimic either too much hormone, you either have too much hormone overtly, or perhaps you have an inappropriate response, perhaps mimicking the presence of a hormone. Or you can have exogenous hormone, like say from a tumor. Or you can have syndromes of deficit of a hormone. Either you lack the receptor or you're not secreting enough hormone in the first place. Maybe something with second messaging down the line, or maybe you lack the response elements to do this. And it's a fascinating topic because hormones, in fact, are one of the major interfaces between the central nervous system and the peripheral tissues. And there are powerful control systems present in hormone signaling. Key with hormones is that they unlike, say, cytokines, we have specialized endocrine glands, sometimes individual cells, that are secreting into an extracellular compartment, usually interstitial fluid, rarely directly into plasma. Usually these things are diffusible enough that they can just secrete into the extracellular space and they'll make their way to the bloodstream just fine. Remember then that only receptor cells with receptors will respond. So if we lack the receptor, we're going to lack a response. Just a reminder that the function of hormones is to either elicit or suppress a cellular function of some kind, which can be growth, movement, metabolism, secretion, any type of cellular function. Hormones probably have a way to either cause more to occur or less to occur. Sometimes it involves enzymatic modulation. Or activation of, like for instance, ionotropic receptors to cause ions to move across a membrane. Or often we associate hormone function with gene expression and protein synthesis. But these ones up here are what we would call the fast cytosolic type responses, and these ones down here would be the slow genetic responses. But it's the same story that we talked about in Chapter 6. We're dealing with one of 2 major kinds of responses, and usually both are going on. To demonstrate the profound power that hormones can have, particularly when you're talking about slow genetic effects, this is the tallest man in modern history. He passed away in his 30s, still growing at 8 feet 11 inches. Actually died of a foot infection due to the neuropathy and vasculopathy issues of his extreme height. More recently, this is I believe, the current world's tallest man. He suffers from the exact same condition. He has a pituitary tumor that secretes growth hormone. It's a benign tumor. It's not malignant, but it's secreting large amounts of growth hormone, triggering his continued growth. It's one of these things that in giantism, in the classic giantism, occurs pre-puberty. The same condition, if it occurs post-puberty, leads to a sort of phenotype along these lines. This was a famous wrestler named Andre the Giant who really came into fame when he was cast in the Princess Bride movie from the late 1980s. But he suffered from the same condition, only the onset occurred post-puberty, which gives his characteristic facial features and truncal morphology different from that lanky one that we see in the giantism. Same condition, just different time of onset. Post-puberty, you have changes in your receptors in your bones and so forth. So more of the plate-type bones in the skull are growing at a faster rate than the long bones do. He still has some long bone growth, but it's not nearly as profound as we see in the giantism. For a syndrome of deficit, we can look to these people right here. These people right here have on appearance, have a female phenotype. They present as women. But when you examine their genetic composition, they are genetically male. In fact, often patients discover they are male, not realizing that they are, because they've had surgery. And what happens is that they go in and they notice that instead of ovaries in the spot where the ovaries are, they actually have testes instead. There was actually a famous episode of the TV show ER, where a young at the time presumed female came in, had to have emergency surgery, and they found testes, and so they had to reveal that she was in fact genetically male. So very, very interesting, and some very occasionally socially perplexing questions when it comes to hormone disorders. And so it's a good idea to know about hormones in order to understand how it's going to impact patients and folks that you're caring for. But hormone issues have been throughout history. This figure here, many thousands of years old, is believed to be depicting goiter, which is a condition often caused by insufficient dietary iodine. And we'll talk about goiter a little bit later when we talk about the thyroid. What's happening is the thyroid is expanding in order to try to increase surface area to harvest any iodine it can get its hands on. Another interesting example from history, a fan of Egyptology, and if you go to any major museum that has Egyptian relics, or if you go to Cairo or British Museum like that, you can spot, this is King Tut's father. So if you're familiar with King Tut, King Tutankhamun, this is Akhenaten. This was King Tut's father. And you can spot him from a mile away because his very, very narrow facial features. And the Egyptians were renowned for fidelity in their figures. So you can identify not only from the cartouches that are found that name the ruler, but also the features are usually very, very consistent. So here's another one of Akhenaten. Again, this long, elongated, narrow face. Or if you look at the full figure, here's what we have. This is Akhenaten as well. Notice the effeminate waistline. And they actually did find his remains, and they did some genetic analysis, and they believe, it's not certain, that he may have had what's called an enzyme excess of aromatase. And aromatase is an enzyme that will convert testosterone to estrogen and lead to effeminization in folks who might have an estrogen excess. It's well understood that can occur. So fascinating stuff, a lot to be learned here. Lots of different tissues that are endocrine tissues that secrete hormones. It's a major, major interface between the central nervous system and the rest of the body. So the heart actually secretes hormones, kidneys, gonads, the adrenal cortex and the adrenal medulla, the liver, skin, fat. There's a hormone called leptin that's released from fat that seems to be associated with body weight regulation to some degree. Lots to talk about, but we don't have that much time. So we're going to kind of cover the highlights and give you a toehold into this major aspect of human physiology. The cells are specialized. They're unlike cytokines. They are specialized cells. They are secreted directly into extracellular compartments and not through ducts. Ducts are, DUCTS, are used to move material to either the external surface of the body, like in the case of sweat glands or oil glands, or into the digestive tract for digestive enzymes, bicarbonate, and so forth. But we don't have ducts in endocrine tissue. Again, only the cells with receptors are capable of responding, but if the tissue has different receptor for that molecule or a different second message pathway present in the cell, not all pathways are present all at once. So if it has a different receptor and or a different second message system, it will lead to that phenomenon that we've talked about called pleiotropic responses. That is to say, different responses in different tissues to the same signal. And hormones follow into the chemical diffusible signal category of our three major categories. Specific cells, I've already mentioned that, right? Transported by the blood oftentimes, sometimes lymphatic tissue, sometimes just through the interstitial fluid. And the targets can be autocrine, can be paracrine, but have the capability of influencing distant tissues, more than, say, 5 to 10 cells in diameter. Very, very powerful. They can activate responses at very, very low concentrations. That's actually one of the classic definitions of a hormone, is that you have to be sensitive to it. Responses have been recorded in model systems with as few as literally one molecule. They have assays so sensitive now that they can actually record responses at one molecule, and that implies a large amount of amplification in the target tissue, as I discussed with, like, for instance, protein kinases or cleavage cascades and second messages. There are even molecules that behave like hormones. They elicit physiologic and or behavioral responses on other organisms of the same species, and they're extraorganismal. Talk about being able to elicit a response at a low concentration. In the case of insects, they will use these things to trap pathogenic or potential pests in crop-sensitive areas. There is some very, very limited evidence that this can occur in humans, but the evidence is very, very limited. That's not to say that it doesn't happen, but it is very much like our little toe. It doesn't cause overt behavioral changes like it might in other species. It may influence decisions, it may influence mood, but it is not necessarily the sort of mind control that, for instance, certain body wash and perfume companies would lead you to believe. So it's nice when somebody smells good, but it's not going to influence the decision you've probably already made about whether that person is attractive or not. And so marketing companies will exploit this. So it's good to know that, yes, there's some very, very limited evidence of that, but not worthy of the major attention that it gets sometimes. We're talking about billions of dollars in odors and things sold every year on the premise that it will somehow influence the opposite sex in some way that they don't have control over. The response mechanisms of hormones are at the cellular level. It may initiate a biochemical response, and it depends on the receptors that are bound. Some of those cellular mechanisms may influence things like neural function, mood, and or behaviors of sorts. And because these things are at such low concentrations and potentially powerful, one of the key concepts when dealing with hormones in the body is this idea of how long the hormone will be present in the blood in a biologically relevant level. And we call that a half-life. How long does it take for half of that concentration of that molecule to be eliminated from the blood plasma? And so it's a logarithmic progression. So 4 hours, we get half, another 4 hours, another half, 4 hours, another half, 4 hours, another half. The idea of threshold in relationship to half-life becomes a very, very important concept. But as a reminder, we're talking about the same pathway as before in those target cells. We have the presence or absence of that hormone, a diffusible signal of some kind, interacting with a receptor, leading to a message or events in the cytoplasm, ultimately activating or altering target proteins that then cause a response. And the responses can be fast in some cases, the responses can be slow. In terms of the structural categories of hormones, we have three major structural categories. We have polypeptides. These are basically small protein-like molecules. Usually they don't form multiple subunit type things. They generally are one polypeptide, like for instance secretin here. We have molecules that are derived from amino acids, sometimes referred to as amines. Tyrosine and epinephrine is a derivative of tyrosine. So we're going to talk about those. And then lastly, we have what are called the steroid hormones, which are derived from cholesterol. So an example of one is a cortisol. We're going to talk about all three. Let's get started with the polypeptide hormones first. Way that we can control peptide hormones is by post-translational modification. That is to say, we start off with a prohormone, sometimes even a prehormone, in an inactive form. And then there are post-translational modifications that lead to the active hormone. And sometimes those modifications occur immediately prior to secretion. So here we have pro-opiomelanocortin, that's a mouthful. It gets chopped up into three separate hormones, at least three. We have adrenocorticotropic hormone, we have gamma lipotropin, and beta endorphin, which many of you might be familiar with. All three of these come from the original peptide sequence, along with this other peptide fragment, which may have something to do with the processing and targeting of this peptide to a vesicle before it's secreted. In any case, we can control this by controlling when these peptides are cleaved and or when we secrete. So it's an overlapping, redundant system of control, so you don't get too much or, in some cases, too little secretion. Here's insulin. Here's the story of insulin. We start off with actually a pre-proinsulin. So here's the original primary sequence of insulin. And then what happens is we process it. The first thing we do is we get rid of that signal sequence and we create these disulfide bonds. This one looks kind of weird, but it's folding the protein in the right way. So we're folding it, and then we wait. So we get to proinsulin here, and this is what's usually residing within insulin granules, the pre-secreted insulin granules. It's ready to be secreted. We just have one more thing to do, which is cleave right here and right here. And that gives us the active insulin molecule, where the disulfide bonds maintain the proper folding and or hold those what were originally different parts of the polypeptide chain together. It's interesting because in some forms of diabetes you secrete the proinsulin and it's not active. So oftentimes what they'll do when they're trying to diagnose a person's diabetes is they'll look at the amount of insulin in the blood, but they'll also look for the amount of this C-peptide. Because by looking at the ratio of C-peptide to overall insulin, they can tell how much of it is actually in active form versus inactive. Peptide hormones are large enough and aqueous enough that they cannot, I repeat, cannot enter the target cells. So they must have receptors in the target tissue that are cell membrane receptors. They can be G proteins in some cases affecting permeability. For instance, if a hormone affects behavior, It can profoundly affect neural pathways by modulating neural ion permeability, or perhaps a receptor enzyme of some kind, leading to either cytosolic or long-term genetic responses. But they're always going to be cell surface, cell membrane receptors and not cytosolic or nuclear. And so that's polypeptides. Again, note the way we can control by post-translational modification and then the type of receptor that it would bind, which would be a cell membrane receptor. Now let's move on to what's probably the most complicated signaling regimen, and that is with the steroids. These guys are highly lipophilic, very, very lipid soluble, and so they have sort of a complicated lifespan in the body and can do a number of different effects on the cells. The steroids, one major area of steroids, gonads can secrete these things like the ovaries and the testes can, but one of the major areas and more diverse areas of secretion comes from the outer layer of the adrenal gland, what we call the cortex. You may have heard of steroids as termed as corticosteroids. That's because they're arising from this outer layer. So the outer layer of anything like the cerebral cortex is the outer layer of the brain, of the cerebrum. The middle of the organ we would call the medulla. This is where we see some amine hormones, catecholamines and things. But this cortex layer is where we see the steroids coming from if they're coming from the adrenal gland. So the middle and the outer layer or surface. So steroid hormones are usually derived from cholesterol, and they are exceptionally lipophilic, easily cross cell membranes and organelle membranes. They are all consisting of this ABCD 4-carbon ring structure. 3 hexagons and a pentagon at the end there, A, B, C, and D. You spot that, you can spot a steroid hormone from a mile away. Here's cholesterol, the precursor. Change the functional groups on that carbon backbone, you can have profound changes in body morphology and, in some cases, behavior. Problem is they're so lipophilic they don't do very well in hydrophilic environments. They are too oily. So how we overcome that is we have special carrier proteins in primarily a form of protein known as a plasma albumin. The albumin proteins have a hydrophobic interior which allow for these lipophilic molecules to diffuse into those albumin proteins and keeps them in the bloodstream longer. They aren't pushed out of the bloodstream by the oil-water situation. So it allows for a longer half-life of molecule and better kinetics of distribution throughout the body, a more uniform distribution. You do see a lot of cytoplasmic and or nuclear receptors for these steroid hormones, which implies more frequency of slow genetic effects. The deeper the receptor is, the more likely it's going to be involved in genetic responses, slower acting. Sometimes the responses may take years or even decades. Gals, if most of you are going to be in the 20-21 year of age range right now. By the time you're 30, your voice will have probably changed significantly. So if you have a recording of yourself talking at your age right now, or maybe a year or two younger, and you hear yourself later on between the ages of 25 and 30, your vocal cords will have matured in response to the estrogen that's been circulating in your blood for the better part of a decade or more. A slower acting response in the vocal cords. Young women who are who are sopranos in high school and college, oftentimes if they are moving on to professional or semi-professional careers, often have to change the parts that they're singing because their vocal cords mature and they're now either a contralto or an alto voice tone rather than the soprano they originally were. You can even hear this across careers in certain singers. There are cell membrane receptors for these steroid hormones. It's not exclusively cytoplasmic or nuclear. So you can get faster, like for instance, neurological responses are faster, non-genetic in nature. So you can get those. Nothing here to really study other than to say that the cholesterol is the precursor. Any number of hormones that we might be familiar with, like aldosterone or cortisol or estrogen, otherwise known as estradiol, testosterone and its close companion dihydrotestosterone. But there's that aromatase enzyme that I was talking about earlier. It can not only convert testosterone to estradiol, but it can also drive the precursors of testosterone into that estrogenic biosynthesis pathway. And steroid hormones are not the sole domain of animals. And the pathways associated with steroid-like molecules transcend species, and we even see versions of this in plants with auxin. Looks like a very similar pathway. What occurs is estrogen diffuses out of the bloodstream, binds with the estrogen receptor. The estrogen receptor will dimerize and then literally grab hold of DNA in certain sequences. We call those estrogen responsive elements. But in any case, there are discrete sequences that are involving genes that are responsive to estrogen. And it will grab hold of that DNA and literally cause the DNA to get torqued to make it easier for transcription factors to bind that region of DNA. They called the estrogen receptor, I saw an article called the DNA gender bender for that torquing action on DNA to make sequences more accessible for expression. But we've, since that first discovery of that physical influence on DNA, we have now seen that occurring in other hormone systems. I'm certain that testosterone receptors do the same thing, just in other sequences, or the other hormones do the same thing. And here's one of Silverthorne's big mechs, and it's basically going through the major aspects of the life cycle of a steroid hormone, as it's starting with where it happens in the bloodstream and then moving into what it can do on the cell. So let's walk through this sequentially. First of all, because that molecule is so hydrophobic, because that molecule is so lipophilic, for proper distribution in the tissues, it really does need to be associated with a protein carrier. There are some syndromes out there where if you take certain types of drugs that may interfere with the hydrophobic interior of an albumin protein or compete for these spots, that you can get hormonal side effects. So it's traveling within its protein carrier, and it's will move on and move off so we're going to start in the bloodstream, and most of the hormone, as it's circulating in the bloodstream, will be found associated with a protein carrier. There will be some equilibrium going on, that you're going to reach a ratio of bound versus unbound. But as molecules are unbound, they are diffusing out of the bloodstream at any given time because they're so lipophilic, which means more of the bound form is going to be released. Assuming we get to a target cell, that hormone can diffuse, if it's free from the protein, can diffuse through those membranes and either interact with cytoplasmic or nuclear receptors to mediate genetic responses. But hold on a second. It's possible that target cell may have a cell surface receptor and interact with it that way for rapid responses. So step 2 is talking about where the receptors might be, and the sky's the limit when it comes to steroids. That's what makes it a little more complicated than the others. If it's a cytoplasmic nuclear receptor, you can almost bet money that it's going to lead to some genetic response, some change in protein expression. So changes in mRNA abundance, changes in mRNA abundance lead to changes in protein down the line. Upregulation, downregulation. And so that's steroid hormones. Again, the carrier proteins, the fact that we can bind cell surface receptors or cytosolic or nuclear receptors. Now let's move into the amino acid derivatives. Sometimes they call them amine hormones and so forth. Lots of small molecules that are hydrophilic, which means that most are not going to be able to get through the plasma membrane. So you can bet money that if it's an amino acid derived molecule, it's going to bind a cell surface receptor like the peptide hormones, but they're much smaller. The main families of amino acid derived hormones are from precursors tryptophan or tyrosine. Melatonin, which folks might take in order to help them fall asleep and stay asleep at night, melatonin is derived from tryptophan. So it's originally metabolized from tryptophan into that. And that's one of the sort of urban myth theories behind when you have a big turkey dinner like at Thanksgiving, you tend to get really, really sleepy. That's actually not entirely true, but you know, there may be some biochemical evidence to support it. It's probably just as much about the fact that you just have a boatload of insulin going through your body because you ate too much. Examples of amine hormones include, like I said, melatonin, derived from tryptophan, the catecholamines, and I want you to look at the similarities between these three. So we have epinephrine, norepinephrine, and dopamine. Probably these two are you're the most familiar with. But here's norepinephrine. You can see it's got a very similar ring-like structure in this tail right here. That's why they're called catecholamines, because this is called a catechol structure. And here's dopamine, very, very important for things like Parkinson's disease and in certain forms of addiction. The last one I have here are the thyroid hormones. The thyroid hormones are derived from tyrosine, like the catecholamines are. The way you can spot thyroid hormones, either there's 4 iodine, so 4 here, that's your classic thyroxine, that's your classic thyroid hormone that modulates metabolic activity in the body. There's also another one, a close cousin, called triiodothyronine. It's a T3, so it has 3 iodines. But those iodines are the giveaway that this is a thyroid hormone. It's the only known hormone in the body that utilizes iodine, and it's made in the thyroid. If you have a lack of dietary iodine, you can suffer from a condition known as goiter. This is a hypertrophy, hyperplasia situation where the thyroid cells have expanded to try to increase their surface area to harvest any iodine they can get a hold of. Iodine is critical because thyroid hormone is a key regulator of rate of metabolism in the human body. If you don't have enough thyroid hormone, you can be hypothyroid and you have slower metabolism where you can lose function. Or you can be the opposite. You can have a hyperthyroid and you're burning through calories and you can't just quiet down when you need to. So it can lead to disorders of behavior and body mass and so forth. That's why it's so critical, and why if you don't have enough dietary iodine, you can suffer from this condition. But we've overcome that in most of the developed societies, in the industrial societies, because what we have done is elected to put iodine in table salt. If you look at a can of Morton's, you'll see iodinated salt. You'd probably have to go out of your way to find non-iodinated salt. They also will sometimes put it into water supplies in order to compensate for low iodine in the water. And that's probably what's going on with this person here, is that they're taking water from a non-industrialized source. They don't have enough iodine in the rest of their diet, and so have developed goiter. It's much more common in agrarian societies, underdeveloped or non-developed countries. We're just hitting the close to the 35-minute mark right now, so I think it's probably a good time in this presentation to call for a pause, and I will pick up with the remaining slides in part 2. Talk to you soon.